Hey friends, happy Halloween! So it's the day before Halloween. I'm getting ready for the big show. You notice my costume. I'm wearing my lab coat, my goggles, which can see nothing. Uh, I'm dressed up as a scientist who studies spiders, an arachnologist. Totally fake, of course. I'm just pretending because what I'm going to do today is I'm going to make a video where I'm going to talk about cuttlefish. Yeah, nothing about spiders at all. Sorry, I know you're heartbroken, but one can't talk about spiders all the time, although my wife thinks I try. All right, so what I'm going to do today is I was browsing through some old websites. I looked at Evolution and News and Views, which you may have heard of. It's the terrible, cheesy website run by the Discovery Institute to promote their bizarre views of evolution. And it was an article by David Klinghoffer, who's a total ignoramus. He knows nothing. Uh, but he was praising these two tweets from Douglas Axe. Doug Axe he's got credentials. I guess he's what counts as the intellectual big guns of the intelligent design movement. But what he wrote was incredibly stupid. It was just plain wrong. It was completely missing the point of everything. So, of course, I felt motivated to sit down and talk at you and explain what is wrong with Douglas Axe's tweets. So we're going to talk about that. And the tweets were about a paper that he misread and didn't understand about cuttlefish Evo Devo. So we're going to have to do a little bit of discussion of deep background. Is that okay? Okay, let's get to it. This is the first one. A find that the axes of cuttlefish limb buds are patterned by the same pathways that regulate arthropod and vertebrate limb development is a surprise to him. But if he'd talked to any developmental biologist in the last 30 years, he'd have known that this was an expected result. This is one of the big stories that emerged as Evo Devo arose in the 1980s, that we find so many common shared developmental programs in diverse organisms that have all just been simply reshaped, reused in subtly different ways to make new structures in different lineages. So, for instance, we use the same genes, the Hox genes, for instance, to pattern the axis of the body plan. And those are found in all kinds of organisms. They're found in fruit flies and people. So we've known about this for a long time. He should not be surprised. He's pretending to be surprised. Either that or he's really that ignorant. Worse, though, he goes on to misinterpret the results. He goes on to suggest that the independent evolution of limbs in cephalopod mollusks involved recruitment of an ancient genetic program for appendage development. In other words, the idea came before the implementation. Okay, the part about the program appearing before the limbs, yes, that's true. You see, the limbs of cephalopods evolved independently, a process that involved recruiting ancient genetic modules. Again, this is no surprise to evolutionary or developmental biologists, but to Doug Axe, who is neither, it means that the idea of limbs preceded the formation of limbs. It's an interesting abuse of the word idea, and is a blatant attempt to infer intent from reuse of genes. It's either dishonest or Axe doesn't understand some common concepts in evolutionary biology. So I'm going to have to introduce you to some of these common concepts. Today, I've set myself an ambitious goal. First, what I want to do is explain to you these three concepts. They're very general, they're easily comprehended, they provide a framework for an alternative explanation to how you could have the genes for limbs before limbs develop. Okay, these three terms are exaptation, bricolage, and deep homology. Many of you may already know this stuff. These really are very common terms in developmental biology, so you may have been exposed to them before, and I'll just give you a brief overview of them. So we'll all be on the same page, and we'll all understand why Axe's interpretation is so bogus. Okay, so after that, what I'm going to do is I am going to give you a brief overview of spatial patterning mechanisms in animal development. This will be a little bit difficult for me. Not because I don't know this stuff, but because I have taught whole semester courses on just this material. And even there, I feel like I'm zooming through it. We have a lot of information about patterning genes, about interactions, about signaling pathways, how all this stuff communicates to build complex structures in organisms. So what I'm going to do is I'm going to shave it down to just a little sliver, a tiny little sliver, and give you an idea of the kinds of things we're actually looking at. So we can stop misrepresenting developmental biology and evolution as the intelligent design creationists do. And then finally, the last bit. Let's look at that cuttlefish paper. So it's a nice paper. It describes the evolution and development of cuttlefish limbs, and it fits completely with the stuff I have said just now I will tell you about. In fact, the authors would probably be very surprised to hear that their work is being corrupted in a misguided attempt to support intelligent design creationism because it is mainstream evolutionary biology. There are no real surprises here. It's just good, solid work to document something that's going on in another clade of organisms, the Spiralia. Okay, you ready? Let's get into it. But let's try to figure out what the heck Axe is talking about here. He seems to think reuse of genetic circuitry in development is evidence of intelligent design. This implies that he thinks evolution would not recycle existing genes, but that if you get a new morphological feature, it ought to appear using only de novo genes. Everything about it must be brand shiny new with no predecessors in the genome. I don't know if this is specifically what he was trying to imply, but there's a common idea in intelligent design called front-loading. They argue that sure, evolution occurred, but it was all simply an unfolding of a complex sequence of programs inserted into the genome by the designer long before they were needed. So early vertebrates didn't have limbs, but buried somewhere in the genome, they contained a recipe for future utility. There was code somewhere in there that would, when the time was ripe, suddenly blossom into limbs fully formed. Some of them even argue that that's what junk DNA is. It's our destiny, our future. But the problem with this is that evolution does not predict that new morphological traits must emerge suddenly, fully formed, with no molecular antecedents. In other words, let's imagine a primitive, limbless bilaterian, a Precambrian worm with no legs. In Axe's view, evolution would predict this worm would sprout legs all at once, and that any reuse of existing genes, such as ones that pattern the anterior-posterior axis of the body, would be cheating. It's a remarkable idea. It just happens to be the exact opposite of what an evolutionary biologist would predict. In fact, if we had evidence that the complex network of genes behind limb formation appeared in one unitary event with no connection to past molecular development, that would be considered evidence against evolution. As usual, creationists walk around with this grossly distorted false picture of how evolution actually works in their heads, and then consider it a triumph if they punch themselves in the brain. It's always opposite day up there for creationists. So how did evolution actually produce limbs? Let's look at the three concepts I mentioned earlier. The first concept that makes a total wreck of Axe's assumption is this one, the idea of exaptations. Evolution is not a linear process. traits do not necessarily evolve in a straight line from a precursor to a specific final function. An exaptation is a feature that performs a function that was not produced by natural selection for its current use, but instead its function is changed over time. A feather is a classic example. Flightless dinosaurs had feathers, so clearly its initial function was not flight. Feathers were initially for insulation or possibly for courtship displays. Their flat shape secondarily made them useful aerodynamically, so they were later secondarily co-opted for flight. We'll see the same thing in limbs. Genes that were initially important in establishing the anterior-posterior axis of the body could also be used for establishing the proximal-distal axis in limbs. It's no surprise when existing gene networks get co-opted for new functions. So exaptation, that's the first thing that Axe seems to have missed. Bricolage is also a common concept. It's taken initially from art and architecture, but Francois Jacob also appropriated it. I guess that's an act of exaptation too. Anyway, it was stolen by the developmental biologists to say something special about how evolution works. You might be familiar with the ideas of collage and found art, where existing elements are organized into something familiar to us, but not at all what the individual components were intended for. I always think of the touristy little stores at the beach when I was growing up, where shells and stones and bits of driftwood were assembled in new arrangements and sold as kitschy little souvenirs. It's amazing how sticking a couple of googly eyes onto anything can make a face appear. As Francois Jacob explained decades ago, nature does not work like an engineer with a plan and a design and a final end product in mind. It's more like a bricoleur, a tinkerer, cobbling together extant bits and pieces to build novel arrangements that have a new function. To make a limb, nature does not start from scratch and build a new feature. It links together existing bits and pieces, gene networks that evolve for a different function altogether. Nature tinkers mindlessly until some arrangement fits a function. And finally, and most importantly, we have to grasp the concept of deep homology. This idea arose from the fact that, for instance, the legs of humans, cuttlefish, and fruit flies are not, strictly speaking, homologous. Our shared common ancestor was a legless worm, and each lineage independently evolved legs, very different looking legs. Yet when you look deeper at the genes involved in developing legs, you find that all three use related genes in similar ways. The explanation is deep homology. Non-homologous structures arose by the modification of pre-existing genetic regulatory circuits. The genes involved are homologous, but the structures that arose are not. Shubin, Tabin, and Carroll gave several examples of this phenomenon. For instance, the eye. The last common ancestor of squid and humans lived roughly 700 million years ago, yet eyes evolved in vertebrates over 500 million years ago, and in the octopus about 480 million years ago. So these are independent events. They weren't generated from a shared ancestor. Fernald estimated that about 4,000 genes were expressed during the development of the eye, and about 70% of them are shared between octopus and human. But morphologically, these are completely different structures, while molecularly, they're using many of the same genes. It is no accident. Our eyeless common ancestor had simple sensors that responded to light. They didn't form images, but they could sense day and night nor shadow passing overhead. They all used opsin, the protein opsin, and retinal as photodetectors. We recruited other molecules like G proteins and enzymes to amplify the signals. And the cells that respond to light, the rhabdomeric and ciliary receptors, were independently assembled into image-forming sensory organs, camera and compound eyes. Are the eyes of an insect, a squid, and a mouse homologous? Not really. But the molecules involved in photoreceptors share a deep homology. And there was a primitive cell that was a simple light detector that we all had in common. But the complex structure of the eye, no, that arose independently in multiple lineages. Here's another amazing example of the reuse of genes. Rhinoceros beetles have those unusual prominent horns. When you examine the genes behind their extension, though, you find familiar limb development genes. Genes like distalis. Distalis is a gene that promotes cell proliferation and seems to crop up all over the place in any kind of bulgy, sticky, outy structure that appears. And aristolis, which is named after the segments of the limbs that are lost when they are mutated. There's also dachshund, dak. That's named after the short, stubby legs of mutant dak flies. These genes that were initially for limb development were simply co-opted for shaping these odd extensions of the thoracic cuticle. There are more surprises. Genes like DLL, distalis, and aristolis have also been co-opted for use in organizing the insect eye and spots on butterfly wings. Why invent a whole new set of genes for the complex task of defining the shape of a novel horn when you've already got a set of handy-dandy genes that interact in a patterned way in the legs? Just turn them on in the thoracic cuticle. They're already configured to form a spatial network. All right, have you gotten it? We're done. Basically, we're done. All you need are those three concepts, and you can slap down Doug Iax's silly idea of some kind of designer behind limb development, because all you got to do is tell him you got an alternative. It could be exaptation. These complex genes were evolved for a different purpose. It could be bricolage, that what happened in evolution was that we took utilities that evolved for one purpose, and we just sort of glommed them together. Well, evolution did, not me, but evolution glommed them together until you got a functional result. Or alternatively, it's just simply explained away by deep homology, that we know about these kinds of phenomena, that there's all kinds of features that aren't necessarily homologous, but they keep recycling, reusing, co-opting the same genes throughout evolutionary history. And that's what we've got going on in these limbs. Now, as I said, we're done. We've explained everything. You can stop now if you want. Just go have fun. Go play a game or something. But I'll go a little deeper into the mechanisms, and we'll talk specifically about some of the genes involved in patterning the limb. Let's return to our friendly Precambrian worm, the last common ancestor of all bilaterians. This animal is not a formless blob. It has some significant properties. It's got a front end and a back end, and there are different structures in each: a mouth in front, genitals in back. It's got a top and a bottom, and a left and a right. It's got organs. I'll use the eye as a proxy for all of that. It doesn't actually have an eye proper, but it does have an eye field where there are some light-sensitive cells. How did it get these basic features? One way is that there were molecular gradients. Any asymmetry in the organism gets amplified by the production of a molecule X at one end that diffuses throughout. So any cell can read where it is along the longitudinal axis by measuring the concentration of X. Lots of X, it's near the head. Little x, it's near the genitals. This is cartoonishly oversimplified. We know in multiple organisms what X is, and it's probably not just one magic molecule that does all the work. In Drosophila, for instance, there's an initial gradient of a maternal gene called bicoid, which triggers a whole complex cascade of signals that interact with each other to first subdivide the animal into broad regions, then initiate segmentation, and then switch on the genes that identify the identity of each region, the Hox genes. It's wonderfully intricate. In my developmental biology course, we can spend weeks on just these processes, which I guess I can't do in a YouTube video. So let's simplify. You know, those Hox genes are the end result of the sequence. We'll just pretend that's all there is. we have a bunch of Hox genes that mark off body regions on the anterior-posterior axis. You and I know better, but we're just going to use this as a shorthand for AP specification, okay? Anterior-posterior specification is more complex than just switching on a couple of Hox genes. There's a lot going on here. Now that we've waved off the complexity of one whole axis, let's consider another one, the dorsal ventral axis. This one is also extremely important. The DV axis specifies where your notochord and nervous system will form, and where your gut will form. In us vertebrates, the nervous system is dorsal, along your back, while your gut tube forms ventrally. In invertebrates, this is inverted, but it uses the very same signals, which I'm not going to get into at all because this is already horrendously complex. It's going to get worse. Have you ever looked at dorsal ventral signaling pathways? Here's a simplified version of the genetic circuit. You heard me right, simplified. In this cartoon, we've reduced the pathways to a few simple components. On the left are genes associated with dorsal fates. On the right, genes associated with ventral fates. You see a bunch of genes, morpholinos, those are interfering RNAs, and salts in red. Those are agents that were manipulated in this particular paper to muck up the dorsal ventral axis specification. Don't worry about them. The key thing is that early Wnt signaling in blue, Wnt is a signaling molecule that activates beta-catenin, that's why it has a pointy arrow, which in turn activates other genes and gene products like cordon, CHRD, and noggin, NOG, glucosid, etc. There's a whole complex of genes that are just listed in a cloud there. And those genes then act to suppress ventral fates by inhibiting genes like BMP, bone morphogenetic protein. And you can see in this diagram, they point to it with a flat-headed arrow, which is a shorthand for suppressive effects. This forms a kind of circuit diagram. This is a genetic circuit that if you ever become or are a developmental biologist, you get to read about all day long. I'm assuming that most of you are not developmental biologists, so let's simplify some more. For this video, we'll just assume it's a simple gradient of Wnt to BMP, okay? Again, you and I know better. This is just shorthand for a complex pathway. Now, I can't resist mentioning for any of you lobsters out there who happen to have tuned in that this is simply upside down relative to the arthropod axis. You'll often see it called the SOG-DPP gradient in invertebrates, where DPP, decapentaplegic, is a molecular homologue of BMP, so there's the same thing. And SOG is the molecular homologue of cordon, so again, they're kind of the same thing. And decapentaplegic specifies dorsal in invertebrates, while SOG specifies ventral. All right. But I said I wasn't going to talk about that. So pretend I said nothing. Just file it away for future reference if I ever want to talk about this stuff. So back to our Precambrian worm. It's even more comical now because you all know that all those double-headed arrows in this simple diagram are proxies for a great deal of complexity. Nightmarish levels of kludgy, arbitrary, seemingly random complexity, which we've boiled down to some simple arrows. I hope you're still with me because I'm about to make it even messier. Let's add a novelty. Let's trigger some localized cell proliferation to create small bumps, incipient limbs. So there's our little bumps, little warts appearing on the side of the animal. These aren't very useful as is. Well, they might have been. In an early fish, for instance, these might act as simple veins to help it be more stable. But otherwise, they need something more if they're to be more than a small wart on the animal's flank. They need axes. something to differentiate top from bottom, front from back. We need new signals for the limbs. Where will we get them? I can't emphasize how important this is. I want you to look at your own limbs. So look there. They are not simple cylinders, are they? You've got fingers at one end. You got a shoulder at the other. They have a proximal distal orientation, and you don't want that mixed up, right? So that's necessary for functionality. In addition, if you look at the hand, you can tell right away there's a thumb and a pinky side. The thumb is anterior, this is posterior. In addition, you've got a back of the hand. This is dorsal, and a palm of the hand, which is ventral. You need these organized in this way for your hand to work properly. Okay? If you didn't have these axes specified, you could, for instance, have mirror reflection. You'd have a palm on both sides or something, which would be far less useful than it sounds. So we've got to add some new level of organization to these little bumps that have appeared in our Precambrian worm. And we're going to have to develop some fairly complex information fairly quickly. As I asked before, how do you think we're going to do it? Back to our ancient bilaterian that has evolved into a primitive chordate and eventually acquires some fin folds. It's also going to acquire some patterning genes. So as I just asked, where will they come from? Will they just invent new molecules and new pathways to do the job? Or will existing pathways be recruited to set up the pattern? And the answer is, it used whatever came to hand. We're going to see a lot of recycled molecules and new arrangements, and familiar pathways will appear again and again. This is how this thing called evolution works. And you knew the answer, if you're not a creationist. Let's just drag in the pathways that work so well in the formation of the body axis and reuse them in our evolving limbs, and bring in other molecules that are involved in other pathways. We'll just assemble something new from the old bits. We're going to engage in a little bricolage here. As an aside, I have to add another important point here. When we reuse that BMP axis, that simple double arrow up there, remember that all those other genes I've hidden away are coming along for the ride. In this way, complexity builds on complexity. We're going to keep on adding these amazingly elaborated genetic circuits to new structures, which means we're layering complexity on top of complexity on top of complexity. Which is yet another reason complexity is not a signature of design. It's all well and good to say we'll just borrow the patterning elements from the body axis to make a patterned limb, but in order to do so, we're going to drag in the whole shared genomic library for that and put it in a new environment with new inputs and modified outputs. Everything gets increasingly intertangled and interconnected. So limb fields are going to be set up with an array of sort of familiar molecules. See the Wnt signaling molecules in this diagram? And also there are others that I haven't mentioned, like the fibroblast growth factor molecules. While I didn't mention FGF before, it's not something new that was invented for limbs. The FGFs are a large family of signaling molecules, often bound to extracellular matrix, and here play a role in specifying lateral plate mesoderm and ectoderm. The Hox genes also get recycled, defining regions of the limb, just as they define regions of the body axis. And another ubiquitous molecule recruited into limb pattern is Hedgehog, or sonic hedgehog, which defines the anterior-posterior pattern. It's high on the posterior side, your pinky finger, remember, and declines in the direction of your thumb. Hedgehog family members are used in patterning all kinds of things. Your lungs, your teeth, the midline of your central nervous system. This is again a case where an existing molecule is drafted to organize a relatively new feature in chordate evolution. When we look at the dorsal ventral axis, we see something very familiar. Look, there's Wnt defining the dorsal side, that is the back of your hand, and BMP defining the ventral side, your palm. As is my habit here, though, I'm going to oversimplify once again. I have to. I've been talking for over half an hour, so here's another crude diagram. But the importance of this diagram is this is all you need to know to comprehend that cuttlefish paper. Lop off a limb bud and a vertebrate, and we can see that the dorsal ventral axis is set up by our familiar friends, Wnt dorsally and BMP ventrally, and the anterior-posterior gradient is set up by our new friend, sonic hedgehog, and only later those familiar Hox genes will join in the limb assembly game. So those are the pieces you need to know for this paper is the Wnt BMP axis and the sonic hedgehog axis. And Wnt BMP defines dorsal ventral, SHH defines the posterior side of the limb. All right, my friends, are you ready? I think you are. You got the basics. You are now ready to enter the cephalopod zone. So let's take a look at this paper. So let's get down to specifics in this paper that Doug Axe thinks supports his claim of intelligent design creationism. I'm sure the authors would be surprised. One of the things the authors point out is that there's a lot of information available about the development and evolution of limbs in 2 of the 3 major superphyla of the Bilateria, the ecdysozoans and the deuterostomes. Many of the ecdysozoa are notorious for their numerous limbs. Think insects and centipedes and shrimp. While there's been a great deal of effort putting into studying the more limited deuterostome limbs for self-centered reasons, because humans are deuterostomes. So for some reason that means we're particularly interested in how we develop. The 3rd superphylum, the Spiralia, have been relatively neglected. The Spiralia include snails and slugs, which are basically limbless, and squid and octopuses and cuttlefish, which are again loaded with lots of limbs. All 3 groups evolved from a limbless common ancestor and later made legs and arms independently of each other. From a morphological standpoint, vertebrate, cephalopod, and insect limbs are definitely not homologous. They're different in every detail, and their last common ancestor did not have primitive stub of a limb that was elaborated in different ways in these different clades. Vertebrate and insect limbs have been dissected in detail, and we know that despite their different origins, they use similar molecules in their early patterning, and also have significant differences. This paper is asking if those radically different cephalopod arms also use the same shared molecular toolkit in patterning. And the answer is going to be no surprise. Yes. That answer is given away in the abstract, so let's take a look at that. Their key discovery is that all 3 axes of cuttlefish limbs are patterned by the same signaling networks that act in vertebrates and arthropods. That is not too surprising. This is another example of deep homology. However, there are differences. They point out that hedgehog is an anterior boundary marker in cuttlefish. In insects and vertebrates, it's a posterior marker. So evolution was tinkering, a limb was being assembled, and this handy boundary-defining gene was co-opted, but different lineages used it to define different sides of the axis. Much of the rest of the abstract summarizes the experiments that were done to show that not only do genes like BMP and Wnt have similar expression patterns, but perturbation experiments show that they have a direct causal role in establishing polarity. And then the big conclusion. These results suggest that cephalopod limbs evolved by parallel activation of an ancient developmental genetic program that was present in the bilaterian common ancestor. Not stated is the understood idea that the ancient genetic program was not there to build limbs, as Axe would like you to believe, but was playing a role in other axis-specifying patterns. So here are the key experiments that were done. The authors have figured out BMP is expressed on the dorsal side of a growing cuttlefish arm, as you can see in panel A. They also used a bead soaked in noggin as an inhibitor of BMP. Remember way back in this long video when I showed you a messy diagram of the genetic circuit in vertebrates, and I told you that Wince would act on beta-catenin, which would then activate a cloud of genes that included cordon, goosecoid, and noggin. So noggin is one of those dorsalizing genes. So they're going to suppress BMP with noggin. They're also going to graft anterior portions of the tentacle to the posterior side in panel D. They knew that Hedgehog was expressed anteriorly, so they're going to generate ectopic Hedgehog expression. They then assessed the effect on arm development by looking at a clear marker, the developing suckers. Suckers are supposed to form on the ventral side of the arm only, and they're colored blue in the photos to the right. What they saw is that noggin would induce the formation of dorsal suckers, which weren't seen in the phosphate buffered saline control, while Hedgehog on the posterior margin would induce mirror image duplications of the arm suckers. This is strong experimental evidence of the role of the genes in defining the polarity of the arm. So to summarize, if you lop off a limb bud in a vertebrate, you get the dorsal ventral axis in response to the Wnt-BMP axis. If you do the same thing in a cephalopod, the dorsal ventral axis is also defined by the Wnt-BMP axis, but it's flipped. It's reversed relative to us. Similarly, sonic Hedgehog is a defining signal of the posterior part of the limb bud in vertebrates. In cephalopods, Hedgehog is an anterior signal. So both sets of organisms are using the same genes to organize their limbs, but cephalopods and vertebrates are flipped upside down and backwards relative to each other. This is pretty darn cool, but really not at all surprising. Once again, evolution is reusing core molecular components in novel ways, but still part of a reasonable pattern. Hedgehog is a handy diffusible signal for forming gradients, and Wnt and BMP are in opposition to one another, making them useful signals for generating polarized structures. So here, enjoy some rotating scans of cuttlefish embryos at different stages while I wrap it up. The main things you need to know. First, animals have a toolkit of molecules that can interact in morphologically useful ways. 2, this toolkit is as ancient as animals are, and all animals share that toolkit. 3, when confronted with similar changes in evolution, like the utility of building a cylindrical, sticky-outy thing with bilateral symmetry, they tend to recruit related molecules to do the job. 4, however, the details can vary. Wnt-BMP can set up an axis in one dimension, but which one is top and which one is bottom, that's going to be a product of developmental historical circumstances. And 5, this is how evolution works. A morphologically useful structure can be slapped together far more quickly by bringing together existing molecular components than by requiring new, unique components, especially when there are many components required that have to work together as an integrated whole, and there's a whole body previously evolved with these elements already present in the genome. Okay? These are obvious and familiar concepts in evolutionary biology, which creationists don't seem to understand at all. If you got it, your next exercise is to try and explain the story to one of your more benighted peers. Share the knowledge. It's how we'll get this stuff out there. Okay, thanks, and I will talk to you later. Enjoy your Halloween.